Okay, amateur videography, but the job will be done. This introduction will be completed. Thank you for your understanding. So I hope this will be the final part of my introduction, the clips. So I was just saying in the other clip that we have all these colonies, as you can see in the picture, but overall my interest also is all these colonies have particular morphologies that we want to describe. If you remember your bacteriology in third year, you were describing, I think with McConkey, I gave you McConkey. So recall those descriptions. What were we describing? We were describing shape, we were describing size, texture, and things like that. So we can just go, I can go to the next slide and show you your results for your 2 agars that you will be looking at with real clinical samples, and therefore you're required to handle with caution. Remember all your aseptic techniques, wear your gloves, they should be disinfectant near you as well. But the view here, please focus on this results table. And these are the variables that you will be studying, your colony morphology. And you're expected to firstly identify what is your agar about. Identify as a group, a small group of about 5 of you, because you will have subgroups for your results. So your small group of 5 or 6, someone should be in charge of informing the team, the Google person, so to say. The Google person will tell you this agar, maybe it is a blood agar, we're looking for hemolysis, is there hemolysis, if there is, how does it look like, and so on. So identify what your agar is about. Is it selective, is it differential? If you recall those types of agars, so identify that will be an important part, as you are also getting the other variables. So they're very self-explanatory, most of them: shape, size, color. And in your leaflet there you have descriptions of what terms to use, okay? Especially with the shape, the size, you say entire, irregular, size, if it's less than a millimeter, you could say small, and so on. But other descriptions that I could emphasize or remind you of what they mean: colony density means when you raise your agar plate towards the light, to a good source of light, is the light passing through or not? Okay, that is colony density. And then cohesiveness is descriptions like mucoid, non-mucoid, and such. Okay, and there are other variables that are not included here. I will show you what I'll draw up on the board, but you'll also be describing texture, which is the surface of your agar, of your colony, sorry. So the surface, is it smooth, is it rough, is it glistening, terms like that. Yes, texture. So those are just reminders of what these different colony morphology descriptions are, and you're expected to fill these 2 tables because you are supplied with 2 agars, so you need to fill these two tables for each agar. Okay, so that was about the table. On my next slide here, I'm just indicating that you should not forget that couches are made alongside your smears, your microscopy smears. So there is that important note, because it is a tool, of course, the non culture method, but they go hand in hand. when you're diagnosing your possible bacterial infection. Okay, and I put a note there that recall your differential staining that you looked at in your third year, your gram stain, which was well practiced. But the ZN stain, it was just a small mention in your practical in your third year. And so I just want to emphasize it here that it's a very important type of differential stain that is regularly used, the ZN stain. Okay, so please take note of that. Of course, we are diagnosing under ZN stain the mycobacterium. But the point is that culture and microscopy go hand in hand. Of course, microscopy is quicker, and so you could have your initial results come in, and then of course you have to wait 24 hours or so for your culture results to have the full picture.